Hey guys.
Hi.
Hey.
My phone's going rogue on me. Hi. So is this whose hemipenes are we grabbing?
Yes.
Whose hemipenes are we grabbing today?
Brandon.
Jimmy Armstrong.
Well, if we combine ourselves together, that would make it hemipenes, so.
There's quad. No, it is hemipenes. Never mind. I'll shut up. I'm going back to sharing.
The echidna has quadruple penes.
Are you for real?
Yeah.
We were talking about echidnas with Dr. Gardner, weren't we?
Yes. I think.
Echidna, I think so. Echidnas are weird animals. Yeah. Okay, hang on. I'm going back to sharing this. Nothing's working. What are we talking about today?
Glow-in-the-dark quail and genetics and a bunch of things that we're going to get educated on.
It's called
Very
the Dinosaur
educational.
Project. This is probably the most sciencey episode we've ever done, everyone. And I know I already said that like last week or the week before, whatever it was.
We keep getting more sciencey as we talk with other people and start learning more things.
Yeah, exactly. Okay, Jimmy, do you want to explain who you are and what you do and what your project is?
Hello, my name is James Armstrong. I go by Jimmy. I am currently a high school student at Ashe County High School, but I am enrolled for the early college program at Appalachian University. I work with transgenic avians, so genetically modified birds. Currently, we've already made glow-in-the-dark quail using a jellyfish gene, and now we are later further progressing to the project where we are putting non-avian theropod traits, dinosaur traits, into quail. Why are we using quail? That's a good question. Let's talk about it.
I'm ready. Let's talk about it.
Let's dive right into it. This is super interesting. Keanu was telling me about it, and I'm like, this sounds really badass. It's a little out of the wheelhouse of what we normally do, but it's still cool.
It's connected. It's connected. And I think this is kind of a neat follow-up to our episode with Dr. Gardner as well. It's just kind of more neat dinosaur stuff. And I have been following ever since Jimmy first posted it, I think. I don't know. I hit the smash button on that Discord server so fast. I don't know. So I breed quail myself, so that's kind of how I found out about it. I breed them for feeders for my reptiles. I eat them. They're delicious. They're just like really, really awesome animals all around for pretty much whatever you use them for. So that's how I found out about this project. But yeah, it's badass. It's badass. That's really all I can say about it, and I just want to follow along and learn about it all.
And reptile people learn a little bit more about genetics because in a lot of morphs, there's a lot of genetic makeup, and that's something that we all can learn from, especially when we're doing pairings and we get more of an understanding.
Absolutely. So how did this project get started?
So growing up, everybody's favorite movie, at least when they were younger, had to have been Jurassic Park. I mean, that's why we all got interested in reptiles, right? You saw the movie, you're like,
absolutely.
Damn it, I can't get a T-Rex. Guess I'm going to have to stick with a bearded dragon for now. One day, though. And then, so it's what really kind of inspired me to get into reptiles. And then a couple years ago, actually, I was reading a documentary on Facebook. It was basically a short play-by-play of this guy that had genetically modified dogs at his house to glow in the dark. And then so I got really interested by that. And then so I started looking into it. And then I fell down the rabbit hole of genetic engineering. And then about 2 years ago, we started the project. We did about a year of research. And then after that year of research, we did 8 months of actual developing the genetic constructs and minor testing. And then 2 months ago, we put that year and 8 months to work. And we injected the first quail eggs. And now we've already got GFP. We've already got GFP quail They're on their second generation now. They're actually incubating. I believe we've got 56 eggs in the incubator right now for second generation GFP quail. And then next week on Wednesday marks the first injections for the tail construct with intent of these birds going to life. These are not the first injections to test out the dinosaur traits, though. If anybody wants to go check out the Discord, about a month and a half ago, we tested the lethality of the arm construct inside of quail embryos by injecting it, because we're using a different promoter than what was originally described in the paper we're basing our research off of. And so this promoter, we're using a minimalist promoter to kind of compact on size and make sure it's only expressed in the arms, well, wings. So apoptosis doesn't occur. So you don't get a pterygium and you don't get the actual wings. So the wing looks like an arm and has fingers. Fingers are claw tipped. All of them are functional. They all have functional phalanges, the whole shebang. And then so we were worried with this promoter that we were using that it could possibly express in the heart and cause heart deformations. And so we tested it out first before letting an embryo grow up to adulthood. We were worried about the health and well-being of our quail. So we always got to test out an embryo first. And then the lethality, none of the birds displayed any heart defects whatsoever. And then so we're ready after the tail to progress with arms.
Yeah, you sent me some of the concept artwork of the quail with arms. And they're hilarious. I think, well, it's not hilarious, but it's amazing. But they have such tiny little wings, you know. So are the fingers going to come out from the very tip and the arms are kind of going to straighten out and move forwards? Or are they going to come out of like the elbow joint, I guess? Like similar to bats, what I would call the elbow, I suppose.
Okay, so with current wing morphology inside of birds, if you look at the skeletal morphology, bird wings don't have a typical joint like we do for how their wings connect to their arms. And the reason for that is all birds descended from a common ancestor that flew. And part of the ability to fly is you need more rotation in your wings so then you can get lift. And then so typically speaking, the reason why bird wings are so tucked back is based upon how the arm sits in the joint. And then so part of this trait that we're inserting is actually fixing the joint so it looks normal. And then so the wings will be a little bit more pushed out, as well as typically the actual tip of the wing, where we think of the tip of the wing, you know how it kind of looks like a mitten? And then so this mitten bit is actually composed of 3 fused fingers. The best way of explaining it is kind of making a gun with your hands. So you have the first 2 fingers that fuse into one. They fuse right here and fuse right here. There's a space right here, and the 2 tips of the fingers are unfused. And then you have the 3rd finger, which is equivalent to a thumb, which is fused whatsoever. That's why it looks like a mitten. And then so what this is doing is, early on genetically, there's a 4th finger, but eventually it kind of dissipates due to apoptosis, programmed cell death. And then so what this is doing is these 2 fingers never fuse. This finger never fuses anyway. And this finger is still there because apoptosis doesn't occur. So you get 3 prominent fingers, all of them claw-tipped, and a 4th vestigial finger, also claw-tipped, on a normal arm-like structure. So if you want to think about how the arms are actually going to be opposed, think about the arms on the velociraptors from Jurassic Park, but instead of walking around with broken wrists, have the arms more tilted in, which is what our concept art shows.
So kind of like this.
Yeah, so like this.
Cool. They could, you know, start to be able to grab things and bring them closer if their arms got big enough.
Yeah.
That's neat. So what are you doing to enhance these genes and like these dinosaur type traits?
So all of the traits we're working with besides enamel are already naturally found inside of the quail. The reason why I say not enamel is because the genes are found inside of quail to produce enamel, like amel and the rest of the enamel complex, but 4 of them are heavily mutated, including amel. And then so the birds don't have functioning versions of the genes to produce enamel. They're called pseudogenes. They don't work anymore. And then so all of the other genes work. They've either just been overexpressed, so it's producing way too much protein as opposed to something in, say, Pongovidiceps or like bearded dragon, or it's producing too little protein as opposed to the Hox gene that causes limb identity that's expressed inside of the limbs and forelimbs of all animals that don't have wings. So every lineage, including bats, whales, everything that you can think of. But inside of birds, it's only expressed inside of the legs. And then so without this identity gene being expressed inside of the wings, it helps feed into the apoptosis and the fusion of the phalanges, creating the glove-like appendage. And then so it's
Wow.
targeting the specific promoter regions and changing how the promoter expresses the gene itself to either increase or decrease in expression to create these phenotypes.
Where's he going? I think it's amazing that you're using stuff that already naturally exists in the animal to enhance these traits. Because I think what most people would just assume is you're, throwing dinosaur DNA into these things, right?
Mm-hmm. So I think the biggest thing that we're really going for with this project, the two biggest things, the well-being of the animals themselves. I care about them quite a bit, and authenticity. So sure, I mean, if you wanted to take dinosaur DNA in theory, you'd want to take a fossil like this velociraptor femur. You'd want to do fibula, sorry, my bad. And then you'd want to go and you'd want to analyze it and do mass spectrometry and look for amino acids. But the thing is, when you have a fossil specimen like this, DNA breaks down at such an incredible rate that you can't get DNA out of it. And then so the next best thing that people start saying is, oh, well, you're going to take alligator genes or lizard genes or snake genes and try to put them into birds. Now, personally, I'm kind of sentimental. And so I feel like if you're putting genes from a different species into the organism, you're not getting closer to the common ancestor. You're getting farther away and you're getting more like a Frankenstein's monster. So by taking genes that are already naturally occurring inside of the animal and modifying them to the way that they were expressed, or at least how we think they were expressed, and getting phenotypes and genotypes that we think or see in the fossil record, we're getting closer to the common ancestor of all this.
And what do you know what that common ancestor is?
So avian evolution is quite perplexing. Originally, it was put that some non-avian theropods, this is going back into the early 60s, non-avian theropods, small troodon-like things survived the KT mass extinction, and then they eventually gained flight, and then with the discovery of things like Archaeopteryx, Microraptor, Quadriraptor, the four-winged dinosaur, with stuff like that, we discovered that the bird evolutionary tree was going back before the KT mass extinction, which is where the big meteor hit and the volcanoes and everything. And so it's kind of perplexing because even then when we were before, all of the fossils were from late Cretaceous China. And so being 70 million years ago, but now ratites, so like emus, ostriches, and most large bird megafauna, they're all put back now 90 million years. So the bird tree is kind of perplexing. We have early lineages of the bird tree like Archaeopteryx, Microraptor, stuff like that, Argentavis, Apster ornis. Pretty much if you play the game Ark, you're set on all the different names. But
I play lots of Ark. I feel like I know what you're talking about.
So I guess what I mean by common ancestor is the common ancestor of all of those avian bird lineages. So to get these more non-avian theropod-like phenotypes, so arms that function, a long non-truncated tail, proper weight distribution along the body so the center of mass is above where the legs are, the hips are at. Stuff like that, a longer neck, teeth, enamel, stuff like this. And then that's what we're going for.
Is there I don't know how to word this. I'll just stew over it and try and come back to it. You said that at the end of your project you were hoping your, I guess, final complete success would resemble a Lemusaurus?
Yeah, so a Lemusaurus, it's not more closely related than other non-avian theropods are. It's actually probably farther off because it's a ceratosaurian. Not to be confused with like Triceratops, but as in Ceratosaurus, a very large carnivore. And then so the reason why we chose Lemusaurus as our goal, if you will, is because it already resembles quail as a whole, pretty similar, both skeletally, niche-wise, and physically. So color, how they eat, where they live. They both live in the same regions of Asia. They both eat, in theory, similar diets because they live in the same regions, except now there's more seeds and flowers and the occasional mammal than there was, I don't know, 9 million years ago. And so it was just an easier thing to start with instead of going with a Triceratops or a T-Rex, because that would be such extensive modification and would take so long that when you go with something that already looks quite similar to your base, it's a lot easier to achieve. Like earlier when we were talking about setting small goals as opposed to large goals, a Lemusaurus is a very small goal as opposed to, say, a Brachiosaurus.
That makes sense, especially like, you know, they're both from the same area in Asia. I think it's what we call China now. I think it's somewhere in there. I don't know. Oh, Japan, maybe. I don't know.
So
aren't they
the quail that we
Caturnix
breed,
japonica?
yeah, so the quail we breed are Caturnix japonica, and then the Lemusaurus lived on the coast of modern-day China. Its name actually translates to mud lizard because all modern-day fossils are found inside of mud flats of what would have been the coastline.
Interesting. We had a couple questions. Corin is asking, how long or at what stage will you be offering modified eggs for sale? I know you mentioned it on Discord, but I wasn't sure when you were offering them for sale.
So let me talk about the modified eggs that we would be offering for sale. We would be offering GFP Caturnix eggs, not dinosaur eggs. I need to make that clear. And then we would be offering them around late Christmas, possibly even on Christmas Day. And so the biggest thing is now is building up a population of these purebred GFP quail. And since we've already got 56 eggs in the incubator and we've got an 80% hatch rate, and that's 80% hatch rate is attributed to the incubator itself, not to the transgene. And then so once we have a large population of chicks going, then we're going to go full ham on producing eggs for everybody to own. So if Kiana wants to have one, if Brandon wants to have one, if I want to fill my bathtub with them and then go snuggle, we can all have transgenic quail. And then so legally speaking, we can only sell transgenic eggs. The USDA does not consider a fertilized egg a live animal for whatever reason that may be. And then so if I send you fertilized eggs and you buy these fertilized eggs and then you put them inside of your incubator, I have no control over that. And then if they accidentally hatch these transgenic quail, you have no control over that. But everybody has to realize that you can't sell these live birds without getting USDA approval. So if you get them, it's not like you can give them away to somebody. I wanted to touch on that.
So since we're in Canada, would you be able to send those eggs to Canada? Because that's a whole
I don't
ballgame.
think so. I think it's a Canadian Food Inspection Agency thing. I could ask Lisa though, because if I could get GFP eggs imported into Canada, I absolutely would, because I like weird things. Because and if a glow-in-the-dark quail is the highlight of my 2020, that's a hell of a highlight, I think. Because my 2020's been kind of crappy lately.
I think it's crappy for all of us.
Yeah. We had another question here.
Okay.
How do you get the DNA and try to do the dinosaur mix? Okay,
so the way we get the DNA, are you talking about like how do we modify the DNA? But so I'm gonna assume it's how do you modify the DNA. So what we're using is we're using a lentiviral vector, which is essentially it's a modified virus that instead of having the viral DNA inside. I know 2020, don't worry. So instead of having the viral DNA inside, it actually contains your plasmid of interest with flanking sites. So then the DNA is incorporated into the virus. And then when the virus goes to infect a cell, it accidentally infects the cell with our plasmid. And then hopefully if it's going for a CRISPR plasmid, it uses ambient expression, and then we'll either remove a section of the DNA, add a section, or whatever it may be. And then so these lentiviral plasmids and constructs so the lentiviral vector that contains the plasmid / construct of interest, we get them produced by vector builders. So each viral construct itself is $1,599. And then on top of that, we typically also get the plasmid itself separate in case we want to subclone into something into it further on. So then we can make a modified version in case something wouldn't say go wrong, but in case we want to make improvements on it. And then so the actual plasmid itself for the virus is about $99.
Huh. So this project is obviously not pretty cheap in the grand scheme of things once everything starts adding up. How do you guys go around getting funding for a project like this?
So when our project first started, we had an angel investor. His name's Adrian Cheney. And he put a lot of funding into the lab, and he still puts supplementary funding every now and then. But at the rate we are in with the project, it's not enough to fund the project to get to the next couple of stages, especially with traits like the tail, arms, snout, etc. And then so the reason for that is because a lot of these traits are going to take multiple generations of breeding, which is it's one of my favorite things about this project. Because typically when you hear about transgenic projects or when you hear about selective breeding projects, they're on such opposite ends of the spectrum and nobody combines both techniques together. So somebody says, hey, I want to get a dog that glows blue. And then so one guy says, hey, I'm going to selectively breed dogs and then test the DNA of these dogs to see who produces an extra protein and selectively breed for one that'll eventually make a blue protein. That's going to take over 10,000 years. Then we have guy over here that just inserts a blue protein into a dog, but it doesn't glow the shade of blue he wants. So now this guy's complaining about why isn't it perfect, and this guy's complaining about why it didn't happen immediately. And then so the best part about our project, in my personal opinion, is combining these gene editing techniques with selective breeding to get the perfect traits or as close to it as possible. And so say for the tail, for instance, or better yet, we'll go with tail. And then so when we're injecting this viral vector into the embryos, we're not going to have embryos that eggs that hatch out with tails first time they hatch out. That would be crazy. What we're going to end up is we're going to end up with quail, and some of them are going to be chimeric. Actually, all of them are going to be chimeric, but some of their germ cells, their reproductive cells, will be chimeric. And the reason why we want the reproductive cells to be chimeric is because if those reproductive cells have the modification of interest and they produce sperm, or if they produce egg cells and we breed them together, best case scenario, which is only like a 1% chance of happening, we'll end up with a second generation homozygous bird for the modification. But more likely, which is only about a 10 to 15% likelihood, we're going to end up with a heterozygous bird, a het. And then so if we take our hets and we breed them together, then 1/4 of a Gen 3 is going to be homozygous, and then 2/4 is going to be het, and then you're going to have 1/4 wild type. And then so then we could even go from there to further selective breeding. It's one of my favorite parts of the project.
Yeah, I like the idea that everything isn't necessarily on a cellular level, that you're changing things, or that's not the right way to word it. Everything isn't super sciencey changing it. Some of it's breeding it, changing it.
Yeah.
Well, it's initially using the science to implement the genetics that you're after, and then using breeding
selective breeding to enhance them further. Yeah.
Yeah.
I don't know. My brain's broken. So, I read on the Discord that you are using chicken DNA. Why is it that you are, or sorry, chicken sperm? Why is it that you are using chicken sperm?
So the reason we're using chicken sperm is not all of the modifications that we're doing we can do genetically, and the reason for that is not that we can't do it. It's not that we don't have the technology. It's just that it would be expensive. So like the silky trait, right? to get the proper CPG site and the promoter mutation, it's going to be 18.9 thousand base pairs. Each base pair to synthesize is going to be 9 cents. So if you want to do the math, it's not that cheap. And so
more than tens of dollars.
More than the amount of money that I have in my wallet on hand. And so selective breeding through hybridization is also a good option to cross over traits. I mean, it's one of the reasons why Coturnix quail and even reptile morphs, when you breed different localities together to produce different morphs, it's one of the reasons why there's so much diversity. And so hybridization and get new traits in a different species isn't unheard of. It's actually well used in different animal hobbies. So some of the traits that we want from the chicken that we need to get into the quail are the silky feathers and the long legs. Silky feathers are obviously found inside of silky chickens. And then the long legs are found inside of like modern game fowl, chamois, ocells, saipans, stuff like that. Most of these are Asian fighting cocks. Insert dick joke. And then so by taking a male rooster and using his semen and the sperminator, we can artificially inseminate a female quail, if not multiple female quail with chicken semen, and then all of the female embryos will die because there's a chromosomal mishap. I think the best example for this would actually have to be savanna cats. So savanna cats, I don't know if a lot of people know how you produce a savanna cat, but you take an African cat species and a domestic cat species and you cross them together, but then all the females either are stillborn or born infertile. Your males are your only fertile ones. So then you have to back cross the males for 5 generations before you can get lineages that are where you can get females that are fertile, not stillborn, and males that are fertile. Males are always fertile, but also increase the fertility level. And then so all of the female embryos that come about from this hybridization, they all die on day 14. There's not a single female quail chicken hybrid that has ever been born, and they've been doing this process since the 60s through artificial insemination. And the reason for artificial insemination is not because a chicken and a quail don't want to get frisky. It's because the size difference is kind of detrimental to the health of the quail.
The quail, yeah. Well, with rabbits, you don't want to let your, continental giant or your giant Flemish breed with your, mini lop kind of thing,
Mm-hmm.
unless the female is the big one, because big boys are bad.
So how do you get the cock sperm?
Okay, so essentially, so you know how you can massage a snake to expose its hemipenes? You massage right above or below the cloaca, or you can even put pressure on it.
Yeah.
And then the hemipenes pop out.
Yes.
He's got his hemipenes get excited. So you can do a similar thing for galliforms. But so galliforms are actually kind of weird in bird lineages because they don't have penises. I know it sounds like it's not weird, but like say ostriches, penises, and then ducks actually have the largest vertebrate penis to body size, which is quite crazy that a
Ducks
duck has
are rapey.
They can also have a 4 to 6 foot long penis depending on the species.
And it's a
Holy
corkscrew.
shit.
Yeah, ducks are like known for like trying to rape their female owners sometimes. They're like iguanas.
Where do they put all that dick?
It's a corkscrew.
It corkscrews up inside of the body. Think
And
like
I
an
think it
extension cord when you
inverts.
roll it up. And then so when you massage the cloaca of the male chicken, he'll start excreting a foam-like substance. This is his sperm. You'll collect it in a small cup like these. Once you collect the sperm, you can then grab your artificial inseminator, push here, pull here, push here. Now you're ready to inseminate some quail. Each time I pull this mechanism, that's equivalent to inseminating 1 female quail. After you inseminate 1 female quail with this, the sperm will keep her producing fertilized eggs for the next 6 days. Sometimes longer. It depends on the variety and also the age and variety of the age. I'm losing it. Age, variety, and how like what season it is.
Yeah, because in like right now my birds aren't laying at all because they hate me, and the supplemental lighting I provide them isn't good enough because they're bitches. It's because they're bitches.
That's quite the process.
Yeah, and I feel like it branches off into like so many different projects too.
Yeah. So, how many roosters do you have to collect sperm from in order to inseminate however many females you guys typically do?
So the amount of semen you collect from one rooster can fertilize 4 quail.
Wow.
That's quite a few.
And then so it takes about 4 roosters to fill this thing, and then that's 16 female quail you can fertilize. And then it's not really that hard, and being that the fertility rate of these hybrids are so like the actual fertility rate of the cross generating fertile eggs from the cross is so low that it's not going to take up so much time. And even then, say if we got 1% of the birds for 1% of the eggs we produce are actually fertile, say we do 1,000 eggs, 10 birds, still not going to be enough to actually take up a significant amount of time. And being you collect semen every couple of days so the male can replenish his resources, if you will, it's not really time consuming. It's more like a task that you occur when you're feeding the birds.
Yeah, like they're such easy animals to deal with, and like to get them to foam up, it's just like you go boop, and the foam up, and you have sperm. Like it's easy, and I'm assuming chickens are just as easy as the quail.
Chickens are more easy.
They're just bigger. Actually, they're probably
Yeah.
easier because they're bigger.
Yeah.
And less stupid. I call my quail suicide chickens. Am I wrong?
No.
These birds will do anything to commit suicide. They will drown in like 3 drops
A
of
centimeter
water.
Yeah.
Yeah. Like they're just desperate to commit suicide.
Oh my God, is that a fox? Let me go run into its mouth. Give it a hug.
Yeah, like literally. It's like, oh, this big hand is trying to grab me. Let me just stand here and be stupid. Why is it that you chose the Coturnix quail specifically? Is it because of where they were from?
So we chose Coturnix quail specifically because of their fast sexual maturation rate. So as we know, at least me and you, that Coturnix quail reach sexual maturity at 6 weeks, 45 days. And so chickens reach sexual maturity at 6 to 9 months, depending on variety, food, time of year, a whole treasure trove of different things. And then so by using quail, you get not only pretty much consistency on when they're going to become sexually mature, but it's also significantly faster. So every modification we wanted to do in a chicken that would take almost a decade, we could do in a quail, and we could accomplish it in 2 to 3 years, if not shorter.
That's fantastic. Like I'm always trying to sell people on quail for whatever they need them for, because of how quickly they reproduce. The eggs are only in the incubator 18 days opposed to 24 or something like that. It's just everything about it is fast tracked.
Which I find weird because quail cost more than chicks to get, and they take half the time.
I think it's just because less people are working with quail.
That could be a possibility too, because chickens are in abundance because there's so many chicken farmers.
Exactly, and chickens also produce a bigger product, like a bigger meat product, a bigger egg, everything. While quail is more nutritious, you need more of them. If I'm going to feed my family a meal of quail, we each need 1 or 2. So
versus like 1 chicken.
Probably still 2 chickens for us. I have a big newfie.
And he's not a dog. Well, that could be debatable.
Okay, that's enough. Be nice. Be nice. Rude. So rude. What has been the highlight of this project for you?
Sounds like a lot.
I think the biggest highlight of the project was, as of right now, has to be a toss-up between when the embryos we injected with the arm construct, when we were looking at the embryos under the microscope, and none of them had heart defects, so we were sure that our construct was going to work and it wasn't going to affect the health of our birds. And the second biggest highlight of the project would probably be, if not the biggest for right now, is when the GFP quail hatched out. They hatched out the day before Labor Day, or 2 days before, I don't remember honestly. And it was just seeing them pip out of their eggs, and then me running to Walmart because I forgot to get a black light, and then putting them under the black light at 11 o'clock at night, knowing I have school the next day. And then just seeing them fluoresce green, especially on their feet and their eyes, their irises were green. It was a sight to be held. It really made the project worth it.
Is it just the soft tissues that glow green, or do the feathers glow green as well?
So the feathers don't glow green, and the reason for that is feathers are composed of dead tissue, just like our hairs.
Okay. Yeah.
So the protein, even if it was expressed in the keratinocytes and then was incorporated into the feathers, it's going to break down by the time you actually get to see the feather, because it's going to go from a blood feather, pin feather, normal feather, if I remember correctly. And then by that point, the protein's already going to be so degraded that
it won't be visual regardless of whether it's present or not.
So
but for soft tissue,
I was going to say, with their bones glow?
Yeah, their bones do glow because there's live cells in it. So their nails glow, their beaks glow, their scales on their legs glow, all of their soft tissue glows, their heart glows. In theory, their blood vessels glow. Everything glows, just the feathers sadly don't, because they're composed of dead tissue.
So if your bird was molting, you would have a pretty glowy bird. A patchy glowy bird. Wow.
So
that's wild to me.
Say your bird's molting and it's got a ton of pin feathers or blood feathers. At this point, there's still some live cells in it, so especially the blood feathers. So the blood feather is going to be little green quills on it.
That's crazy.
That's super cool.
Yeah. I love this project. I'm so about this. Everyone go check out their Patreon and support this project if you're interested in it. And support our Patreon too.
Yeah, they're hosting me. You guys got to support them.
We're cheaper than he is. I'm kidding. But yeah, awesome. I think anyone that's interested in this project should absolutely check out the Discord server. You guys post so much information on there. Lots of updates with the project, pictures, stuff like that. So check out the Discord server in the description.
Yeah,
get cool. Discord.
I don't even know what that is. Kiana keeps telling me about it, and I'm like, I don't know what that is.
I don't use it very much, but you can do audio chats and shit. I don't know. You can do stuff.
Stuff and things and
it's like
sciencey things.
It's like fancy MSN with weird chat rooms. Let me age myself.
It's like online communities, but then each community you get to create, and then so you can have specific communities. So different YouTubers have their own personal Discords, and it's kind of like, how you have Facebook groups with all the soccer moms, and they're like, oh yeah, Billy did this today, and then everybody's so supportive. It's kind of like that, but all about the community you create.
We could start a Discord server for the podcast. you know,
Yeah.
and like give our Patreon supporters access to the Discord and have a chance to live chat with us there. But like, I've fucking talked to you enough.
Yeah, that's right. I have to talk to you over Messenger and then on here, and then I have to go back to the recordings and hear your voice again.
And then we just like video chat to video chat it like twice a week.
Yeah.
Let's test audio.
Does this sound like shit?
Talk again. I don't know. It's like going to the optometrist is like A or B. A or B. They both suck.
It's like the Verizon commercial. Can you hear me now?
We're usually a Verizon commercial.
Yeah.
How long do you anticipate this project to take?
Honestly, it depends on success rate and failure rate. Typically, I would let. Still trying to sound humble, I would like to say I'm kind of lucky with a lot of the things I've done. I could have had a lot more failure than I have so far, and I probably will have a lot more failure in the future. But even with a 1% success rate for a lot of these things, the longest it should take is about 4 to 5 years. Shortest it should take is 1 to 2 years.
That's amazing.
That's a lot quicker than I thought it would take.
Yeah, I thought you were like looking at like at least a decade-long project.
Well, so quails can go through 4 generations a year, right? So even if we mess up 1 generation, then we can go through another 3, and then just have to go 1 more generation in the next year to be at the same point we were. So it's really not that setting back if we lose a whole generation like it would be with chickens. Because then if you lose a generation with chickens, then you have to wait another almost 2.5 years to get back up to the point we were at.
Wow. Quail are superior.
So when you guys were first thinking about this project and stuff, what did you guys think your success and fail rates would have been?
So I have a close friend, and he kind of been a little bit of a mentor for the project. He actually works with the project. His name is Andreas Stummer. He's from Austria. And then so one of the things he told me when we first started this project was, Jimmy, whenever you think you're going to succeed, expect to fail 8 times, expect to fail 20 times over that, and then expect to fail 1 more time. And then only then will you possibly succeed. So I had really low expectations for the project. And it's not that I don't believe in myself. It's just I don't want to overthink about my abilities or overput faith into the project and then get disappointed. I would rather think that the project probably won't work the first couple of times we do it, and then if it does, be surprised and be happy, and then expecting it's going to work, and then when it doesn't, I'm upset.
So you mean you're not counting your quail before they hatch?
No.
Ha! No one thought I was funny. Okay, I'm not as funny as I think. I try.
Well, yeah, especially with a lot of science experiments and playing with genetics, not everything's going to take right away, and there's a lot more factors into it too than the general person thinks about. So you guys have a lot of things to take into account for. And there's always a lot more failures than successes.
It's one of the reasons why we wanted to test the lethality that we were using the new promoter. I mean, imagine if we, as I said earlier, jumped the shark and we had just we'd immediately put that construct into embryos, and then the embryos grow up fine, they're chimeric, they produce heterozygous offspring. We're still not at a point where this could be detrimental. And then we breed them, and then 1/4 of our chicks die, all of them have arms. We don't know why, and then we start dissecting them, and then we all realize that they all have a similar heart deformation. Imagine if we did that. Well, that's not the case, and there were no heart malformations or anything. The construct works as intended, but if we had an issue like that without doing any testing, it could have been a major setback. We could have put half a year, almost 2 years worth of half a year to a year's worth of research, and then it would have completely went down the drain. So part of our biggest thing is doing testing first to make sure we don't have foresights that we kind of skipped over. Does that make sense?
No, it does.
Yeah.
There's a lot of sciencey jargon that I am not sure what goes into the whole process, but it's not just throwing genetics into something and hopefully it works. There's there's a lot of science and math and calculations and figuring. Hope this might work, or if that doesn't work, this might.
Okay, I have a question. Okay, you said that you were inseminating quail with silky chicken sperm, right?
Mhm.
And all the female embryos would die on day 14, so you will only
Mhm.
have males that hatch,
Mhm.
and they will all be infertile, right?
Yeah.
Okay, and then you said that you were looking for like the longer leg traits from different types of birds, right?
Hmm.
So obviously you would follow that exact same process, but
Yeah.
and then how do you combine both of those into one if they're both infertile?
Okay. So, did I explain why they were infertile?
I don't
I
think
think
so.
you, not since we hit record. I think you did before we were recording.
Okay. So the
Maybe.
reason that the hybrids are infertile is because, as we've talked about multiple times in this, quail reach sexual maturity so much faster than chickens that the sex hormones, or the genes that produce the sex hormones in the hypothalamus and in the pituitary gland, are saying, "Hey, we need to become sexually mature at 6 weeks." And then the chicken set of genes is saying, "Hey, we need to become sexually mature at 6 to 9 months." And then so this not chromosome, this hormonal mishap is going on, and it's essentially causing the testicles to either become malformed or undeveloped, and then so the sperm is not produced in a high enough quantity for the bird to be able to be fertile. I mean, it's still genetically it's fertile to be able to produce offspring, but there's not enough sperm. So there's a couple different ways you could solve this. Two, actually. You could do hormonal treatments on the birds from a young age on the hybrids to basically get them to either follow the quail's sexual maturation rate or the chicken's sexual maturation rate. And then it would form testicles that are somewhat normal, that would be able to produce enough sperm that if you were to use artificial insemination, you could probably get some embryos. But that would be crazy expensive and also extremely tedious. It would require frequent injections of sex hormones. It would also require frequent vet, and then these sex hormones aren't cheap. So that option got thrown out. The other option that you could do is a germline chimera. What is a germline chimera? So you could take germ cells from a hybrid, whether you actually want to dissect the hybrid itself and remove testicular tissue and then culture it and then implant that into a 51-hour-old embryo with surrogate egg cell culture, or if you want to take one of these hybrid males and you want to remove a feather during molting and you want to culture some fibroblasts and then convert them into a stem cell like state and then turn those into germ cells and then plant those, either way you're going to end up with a chimeric bird. The whole rest of the bird is normal. Its testicles, though, are entirely composed. If you use a chemical called busulfan, if you inject the busulfan, it'll kill off the native quail germ cells. And then so after you inject the busulfan, you wait a couple of hours, you implant your germ cells, and then the quail's germ cells, so its testicles, will be entirely composed of the hybrid germ cell. So when the bird goes up, its hypothalamus and its pituitary are only producing quail sex hormones at quail sex time. So then the testicles get proper hormones at the proper times, will form normally, and then will produce sperm. And then you take this bird, you can breed them to normal wild type female quail, no need for the sperminator. And then you'll get your F3, which is one back cross. Well, no, F2 because it's a back cross. And then I want to continuously back cross for another generation. And the reason for that is I just want to bring up fertility rates in the birds. So just like when we were talking about with the savanna cats earlier, the males will produce offspring, but the fertility rates are quite low. So you might have to go through a couple different pairings to actually end up with a group of kittens. And then so with these, I might have to put them in with a dozen females to get 4 of them pregnant or ovulating. And so just to back cross a couple of times and then PCR the birds for the ones with the silky mutation, just to identify who's heterozygous and who's wild type. So it's not, a wild goose chase. I don't think, well, this guy probably has it and he doesn't. And I'm 6 generations down the line wondering why he's not throwing me silky quail. And so
I want silky quail more than I want glowing quail.
silky quail would be cute. And since they're not transgenic, obviously we could send those out to anybody.
I still have to check with CFIA because it's livestock going over the border. It's probably like an import process.
Probably. And then, so, I mean, once you go through that process, you're essentially going to end up with a bird that phenotypically looks remarkably like a quail, if not indistinguishable. But it has your chicken trait of choice as long as you're using your PCR confirmation to select for your certain chicken trait. In silky chickens, we're looking for the PDSS2 mutation inside of the promoter, which is 103 base pairs up from the CDS coding site. And then in the long-legged chicken varieties, such as the asil, the saipan, the shamo, the modern game fowl, we're looking for a mutation inside of IHH, Indian hedgehog. Yes, named after the Sonic Hedgehog pathway, which is named after everybody's favorite hedgehog.
Yes. Scientists being nerds. I just want to watch this project grow. Like, it's so cool. Like it's so cool, and it's one of those kind of not see it to believe it, but like I can't wait to see it. What are you planning on doing to do the beak modifications?
So remember earlier when we were talking about gene expression towards the very beginning?
There's a comment that says it's not rocket surgery.
Thanks,
For me, yes.
It's brain science, anyway. Part of the beak modifications is there is a gene that was typically expressed. It's expressed inside of us. It's expressed inside of your hog noses. It's expressed inside of pretty much everything with teeth. And then so this gene also helps form the integument on our arms, so our hair, our fingernails. It's part of the EDA pathway. And then so this gene also helps form the buds that will eventually become our teeth and the roots. And so this gene is not expressed inside the mesenchymal tissue. It's a hard word to pronounce, so excuse me. I know I sounded like I was trying to summon a demon, but the mesenchymal tissue inside of the mouth of the bird, which eventually forms the teeth, it's not expressed there anymore. It's lost a section of the promoters called an enhancer. It enhances expression. Very hard to figure out. And then so essentially, since it's lost this enhancer, it's no longer expressed, so birds don't form teeth. But this enhancer also, with unintended effects when it was originally discovered, it also does a secondary thing. It causes the FGF8 and BMP4 expression to change. FGF8 stands for fibroblast growth factor number 8, and BMP4 stands for bone morphological protein 4. And then so when both of these are responsible for head shape, and then so when it changes this expression, you get a crocodile-like snout on a bird. This was originally done in 2016, followed up in 2018, was then followed up in 2019, and then the 2020 paper, which we're basing our research off of, is the most latest paper. In all of them, they were able to produce alligator, if not dinosaur-esque snout on a chicken. And then so we took it, we modified it for viral vector, all of these things. They were topically applying a virus, which is just you coat the tissue of the embryo with a virus, but they have no intent on making the birds able to pass this on genetically. So we adapted their protocol to our protocol, so then it would be able to be passed on, along with making a couple other modifications. And then boom, now the bird will form a snout. In theory, it will form small teeth buds, but being it won't have enamel, the teeth won't progress past a couple of days. Once it starts eating food, teeth will get worn away, which is actually kind of funny. Do you remember when we were talking about the Lemusaurus earlier?
Mm-hmm.
So one of the funniest things about the Lemusaurus is it also didn't have enamel. And then so as it would get older and it would eat, it would grind down its teeth until eventually they were no longer there. Which, funny enough, is exactly what's going to happen if we do this snout modification inside of the quail, because they don't have enamel. Now, whether the same enamel mutations that cause the enamel genes to not function inside the quail, the same ones inside the Tyrannosaurus? I don't know. We probably will never know. But it is kind of funny how just the reactivation of this one gene creates a similar craniofacial morphology and a similar type of, I guess, it's not pathway, developmentary change. I guess
Same,
is the best way of putting
same, but different.
Yeah.
So, with the snout change and then the lack of teeth as they would wear out from eating foods, what would they be able to eat at that point? Because I don't think it would be the normal food that they're used to doing with their facial structure in their mouth.
They eat
Well,
a
no,
pelleted diet generally
Yeah.
without teeth, so that they would be able to eat essentially their normal diet.
Okay.
Yeah, everything stays the same. I mean, personally, since you're going to have different craniofacial morphology and the size of the skull and the maxillar bones are increased, I would recommend probably increasing the calcium in the diet a little bit more, just so you don't have weak bones and stuff like that. You know, got to drink your milk, got to eat your spinach, Popeye arms. And so just stuff like that, so you don't have to worry about the birds having like a weak skull or anything. But even then, it shouldn't be detrimental to the birds. I would just want them to have strong bones, especially if I'm making these modifications. I feel responsible for the well-being of all of these birds. Their well-being is my responsibility.
I think one of the biggest things that these tooth buds would probably do is, if you're giving your birds greens and stuff like that, they may be able to shred them up a bit better. That's like the only real application I can really see for it, because most of us are raising on like a commercial pelleted diet that has all the nutrients that they need, and we just give them like greens as extras because we love the little suicide chickens and don't want them to drown themselves in water.
Yeah. And I mean, if you raise your birds on the ground outside, typically you might have a small lizard stumble into the pen and never return, or small insects that will come in that will supplement their diet. So I guess another application is if you've got like a beetle problem or something.
Oh yeah, they'll eat slugs and all that kind of stuff. I thought about originally putting them in my garden to deal with pests in there, and then I realized the quail will eat everything. Yes, but what's ironic is the bugs ate my garden, so I still got no garden. I would have been better off feeding my damn quail.
If you would have fed your quail, then you would have had not only more quail because they would have been reproducing, but your quail would have been bigger when you ate them.
Exactly. I just sacrificed the garden to the quail, let them eat all the bugs, and then eat all the quail.
And you obtain the essence of the garden.
I am the garden.
And to gain the quail's courage, you must eat the heart. Yeah.
The quail's courage? That's just blind stupidity and fearlessness due to stupidity.
Isn't that what courage is, though?
It might be. Too stupid to be afraid. This seems like a quality I have.
No comment.
Yeah, that's okay. You can comment.
Are there any more comments on either the YouTube or Facebook Live? Curious.
I haven't seen any. Just Brandon's dad joking about getting arrested.
Yeah, and saying chickens are the closest thing to dinosaurs.
Also, Corin left a comment saying quail eggs have more healthy proteins as well, opposed to chicken eggs. Very nutritious. I believe quail eggs have 3 times the nutrition as a chicken egg, and it's like tiny, itty bitty.
I feed quail eggs.
Yeah, that big.
Eggs.
Yeah, you mean a dinosaur egg?
Yeah. I feed dinosaur eggs to my relatives to dinosaurs.
But no, the reason why they're so much more nutritious is because
Corin says if you grew tatsoi, sorry if I'm butchering that, it may help supplement the calcium requirements. Tatsoi is a spinach kale-like plant that has double the calcium as milk. Sorry, my tongue doesn't like that sentence.
I didn't know that. I'll genuinely
I
look
didn't
into
either.
that.
Yeah, no, and, excuse me. And like it's great to be able to like supplement their diet with you know, greens that are actually going to be good for them without causing like weird imbalances and stuff. So Corin, you're smarty pants. Keep it up. Yeah, if anyone else has any more questions about the Dinosaur Project or what Jimmy's doing, comment them and we can get them sorted real quick.
Yeah. So when you guys are doing a lot of testing for this project, what kind of machines and what kind of processes do you guys use?
So a lot of the time, the testing can be done with a simple dissection. So what you'll do is you'll take the embryo, you'll let it develop into a certain point after injection, and then so you're actually one of the machines for injection. You actually have to use a micro injection apparatus, and then another machine that goes along with the micro injection apparatus is it's actually a puller. So it'll take these small glass tubes and pull them apart and form really very tiny needles, and the tip of the needle is enough to puncture a single cell. And then so depending on how far apart you pull the little tubes, you can make different size needles. And when you cut the tube and to form the needle, they're very sharp, extremely sharp. So you can penetrate a cell without popping it. And then so when we pulled needles and everything, or you can even buy pre-pulled needles online, but they're not as good, that requires a machine in itself. But then the micro injection apparatus is essentially kind of like the little air stones you get for a fish tank, but you can gauge in pressure. And then the pressure, essentially, you can either suck in one way, suck in the nucleus, or suck in DNA. And then you could either blow it out, insert sex joke now. And then so once you micro inject your DNA construct or your lentiviral vector into the thing, then you close up the thing with parafilm, and then you put it in the incubator, you let it develop. We're actually letting our GFP controls that we're using because we're doing a lentiviral vector for the tail construct, which begins on Wednesday. So we're testing out and gauging how much virus we need to inject it on what stages to see which would be the best way to produce with the highest success germline chimeras inside of the quail. So for this, we injected 48 eggs using the microinjection apparatus into 24 were injected into the blastoderm freshly laid eggs. Another 24 were injected into the extra circulatory system of 48-hour eggs. So that's HH stage 0 and HH stage 12 to 14. HH stage stands for Henry Hamilton, and it's just a way to gauge different embryonic development. And then so after that, when we gauge different embryonic development, and then we let it develop for a week, so now we're in HH stage 20 something. Then when we cut open the egg and we look at the embryo and we put it under black light, if the wings are glowing, that's good. But what we're really looking for is how brightly and how much of the testicles glow. So say we had like the blessing of all divine deities and the testicles entirely glowed on all of the birds. We would know 100% to go with that method. But I mean, the only thing you really need for that is a inverted fluorescent microscope, which are only about 2,000 to 3,000.
Only.
You know. I don't know if that translates into Canada. I mean, you guys don't have pennies, so.
With the exchange rate, you can almost double it with the current exchange rate.
I think the exchange rate got somewhat better, but
did it? I don't know. I know Brandon and I get screwed on the exchange rate for like all our hosting fees and stuff.
$1 US is $1.31 Canadian.
He's not in Europe. He's in the US.
But I said USD.
Oh, I thought I said Europe. I'm sorry. I wasn't listening.
That would be a euro.
Yeah, it would. Sorry, I was turning off my humidifier because it's turning into a sweat lodge in this room.
And $1 Canadian equals 76 cents in the US. So
sad face.
We're doing great over here.
Have you faced any kind of backlash for this project?
Yes. We've had a couple of people say, well, we've had different types of backlash, which is a thing that happens with every major project. I mean, they're having backlash now on the different coronavirus vaccines, which I'm not going to touch on. But
We're good.
Yeah. So some of the backlash we've had is we're playing God by doing this. My favorite response to that is, are we really playing God if God gave us all the tools?
Ooh. Bazinga.
And then another type of backlash we've had is, where are they going to go? Like, if you make a dinosaur, what's going to happen to it? And then so honestly, it's going to be kind of weird to say, but I like that there's backlash like that. I really do. People care enough about the project to genuinely be concerned about where these animals are going to go after the project is done. Backlash like that I can get behind and I can support. And I would, even if I wasn't the person leading this project, even if I wasn't a part of this project, I would be part of that backlash. And the reason for that is, say we make these animals. What happens to them? Do they go live off on an island somewhere? Do we sell them? Do we put them in a zoo? Is that ethical? Is the project ethical if they don't have a wildlife environment to exist in? And then my answer to that backlash is, yes, it's ethical. It's ethical for the same reason that owning a Chihuahua is ethical. It's ethical for the same reason that owning a German Shepherd is ethical. And then they say, well, how do those two things relate? Well, I say, well, we're making it technically for scientific purposes and for not personal amusement, but as an attraction. And then they say, oh, well, then that's not ethical. And then I say, well, what's a dog breed then? And they say, well, it's a companion. And I say, well, I've never seen a wild Chihuahua, nor have I seen a wild pug. So it's almost like that breed was entirely made for human enjoyment or as a commodity. And so it's no different. And then they say, oh, well, are your birds going to have, oh, well, this is the third type of backlash. Are they going to have health issues? And if so, I mean, it's not even are they going to have health issues. A lot of people think that just because they're modified, they're going to have health issues. And then so when I posted the GFP birds the first time we posted them, I had some backlash with people saying that they were deformed. And then so I posted pictures of the chicks because this one lady, I think her name was Robin Helms, was spouting off about how they have 18 legs and tentacles and I'm the son of Satan. And it was interesting to me. And so I posted a picture of the newborn chicks from the outside of the incubator. I'm like, as you can see, guys, tentacles, the whole wazoo, goat horns. This one's even smoking a doobie, can't you tell? And so, besides stuff like that, I mean, I can support backlash about where they're going to go because that's a legitimate question. When it comes to backlash, like, oh, they're going to be deformed because they're transgenic, honestly, you can have an opinion. I'm not telling anybody they can't have an opinion, but personally, that opinion doesn't seem justified, especially when you say, oh, well, it's transgenic, it's not natural, as opposed to the extremely natural pug that has an entire genetic disorder named after it because its skull is too small for its brain, so it dies prematurely because its literal brain case crushes its brain and dies from too much chronic pressure on its brain. But, as opposed to my glow-in-the-dark quail that have no issues and are on their second generation.
Yeah.
Well, I think your glow-in-the-dark quail are no different than a glow-in-the-dark axolotl, other than they're a different species.
Yeah, or like...
one's an amphibian, and one's an avian. Yeah. But it's the exact same protein, and I don't see anyone saying GFP axolotls are unethical. Maybe they did when they first came out.
I mean, it's kind of like arguing, say if I had a GFP ball python, and then somebody's trying to argue that spiders are better than GFP snakes. And spiders have their own form of madness going on. Yeah. And then so it would be like trying to argue something that it legitimately has a genetic disorder is better than something that's perfectly fine, except it contains a single jellyfish gene. And I'm imagining... Go ahead. When we start inputting other traits, we're going to get more backlash, and I'm okay with that. I don't mind criticism. Just, I do hope for criticism that's well-backed, either scientifically or ethically.
Educated criticism. Mm-hmm. And not just, this is stupid
feelings.
Kind of stuff. Yeah, exactly. Because realistically, feelings aren't involved in this. This is science. There aren't feelings in science.
Science doesn't give a shit about your feelings.
I want it to work, but that doesn't mean it's going to. I mean,
Exactly.
Yeah, like you of course are going to feel positively towards the project because you want it to succeed, but that's not going to affect the outcome. It may affect your work ethic, but it's not necessarily going to affect the project itself. Outcome.
Yeah.
What is transgenetics? I wanted to ask this earlier, but it totally slipped my mind.
So trans means to move across. It's a Latin word. Genetics, meaning the study of DNA or genes. And so transgenesis is moving a gene from one species to another.
Okay.
So that's almost like, well, that's essentially genetically modifying something, correct?
Yeah, they're interchangeable.
Oh, okay. So transgenesis is GMO, I guess.
GMO quail.
Yeah. GMO quail.
Well, yeah. Transgenesis is a form of genetic modification, but not all genetic modification is transgenesis.
Because isn't line breeding also a type of genetic modification as well?
Any form of breeding for a certain characteristic is a form of genetic modification.
Perfect. Everything we do is basically genetic modification.
If you want to only breed anaconda hognose snakes, then you're gene modifying your snakes. Now, it might be eugenics, and that might be its own rabbit hole, but
Someone tried to start a eugenics in reptiles conversation a couple of weeks ago, and it got squashed pretty quickly. I was interested to see how it was going to go, but it got kiboshed very fast.
Well, if you want to know more about reptile genetics, I know a person. Well, I've heard him talk on numerous podcasts about reptile genetics, so we could get him on.
Let's get them on.
it's
I'm here
Brian
for all the
Barczyk.
learning. Pardon?
Gets Brian Barczyk.
No.
No.
No. We do not support crying Brian on this show.
I'm kidding.
No, it'd be Dr. Travis Wyman, because he does a lot of ball python breeding, but he's a geneticist, so he understands all that stuff.
That's really cool.
Yeah.
Yeah, I'm just holding on to the seat of my pants for this live stream. Smile and nod with the fancy terms. I understand the basis of it, like just not the science, super sciencey stuff.
Well, like the more I listen to like actual scientists talk about like the different testing that's involved with all the genetics, and then like the virus testing that's going on within the different reptile species and other things that it applies to, I'm learning a lot more, and this obviously broadens my knowledge on the subject.
You didn't have a choice about doing this. We were doing this.
It sounded cool to me. Like, it might not directly apply to our field that we're involved in, but it still applies to it in some aspect.
Mm-hmm.
You guys want to learn an
so.
interesting transgenics fact about quail?
Yes, I do.
You know what celadon quail are, right?
Yes. They carry a gene that produces blue eggs.
So, funny thing. All birds carry the gene to produce blue eggs, and it's not really a gene it's not that all birds don't have the gene, it's just if the gene turned on or not. And then so chickens carry the gene, that's why you can have blue egg laying chickens and white egg laying chickens, it's if the gene's turned on. And then you say, Jimmy, well, how is the gene turned on? Well, first I think I should explain how essentially it's an enzyme binding factor that binds biliverdin. So the chemical that makes your bruises purple and blue and green, that chemical, it binds it to the eggshell, that's why they're bluish green. And then so basically what this enzyme does is it's turned off in most birds. That's why you'll have white laying quail eggs or quail that'll lay eggs like this, or you'll have chickens that lay white eggs. But a virus, a naturally occurring virus, I think it's avian respiratory virus 40, has a neat little thing where it likes to insert part of its genome just upstream of this gene, and then it'll eventually pop out later on. It's called shedding. And then so sometimes part of the viral genome is left behind. And if that part of the viral genome has a promoter in it, it'll promote the gene expression of this inactive gene, causing this dormant gene to turn on, producing blue eggs.
So your birds get a cold and their offspring makes blue eggs?
No, it's just like every couple hundred times this virus will accidentally leave a little bit of its DNA in the genome of the bird. And then so if it leaves a little bit of its DNA in some of the sex cells, and those sex cells become sperm or egg, and then those hatch into a bird. That's how we got traits like Celadon and Americana and Ericana. It's just natural bits of genetic modification that's called horizontal gene transfer. The genes are being horizontally moved from one species to another.
Huh. That's very interesting.
Yeah.
When we were talking yesterday, I think you were also saying that the Lemusaurus also has that that word
Blue
that
egg.
starts with the blue egg, but what was that B word you said?
Biliverdin.
That one.
Yeah.
So the Lemusaurus also laid the blue eggs, which I thought was fucking cool.
That is cool.
So most dinosaur species that laid hard-shell eggs laid a colored egg. You can see this in a lot of bird megafauna. So if you look at like all ratites, all of them laid colored eggs. The reason for this is other proteins inside the eggshell will bond to it, so they fluoresce under black light. So the eggs blend in with the environment, but to the bird parents themselves, they stick out. They're either bright pink or bright red, or bright orange. And then so colored eggs that blend in with the background that we can't see or that we might stumble past, to us, they blend in. To the birds, it's like, my babies are here.
Wow.
And then so it's a feature to find your kids so you don't lose them.
That's cool. I need to put a blinker on my kids.
Yeah, that's a cool adaptation that probably some other animals have made too. So
one of the reasons why a lot of birds have lost it is you can see a lot of flying birds don't have colored eggs. I mean, look at all parrots. If you look at parakeets, pigeons, stuff like that, where they have to constantly take care of their young and they know where their nest is. That's a lot different than an ostrich or an emu that finds a small divot in the ground, lays its eggs there, and will come every couple of hours after going eating and grazing. If you have to constantly take care of and check on your eggs, you've got to know where they're at. So it doesn't matter if they have color or not, you're just going to remember the spot. Whereas if you go and you walk 16 miles this way and then now you're in the general direction and you're looking for your nest, if you can't see them, they're not going to hatch. So it makes sense evolutionarily the eggs that were the most visible to the parents were the only ones that hatched, and then so on and so forth until the whole population acquired these traits. And if you don't need them anymore, these traits were eventually lost.
It does make sense. I just like it. That was never even like a blip on my radar. What about like robins, because they lay a colored egg?
So one of the reasons that robins lay colored eggs is so they can identify robin nests from other nests, because robins have pretty poor vision when it comes to most bird species. And then so I experienced this, and it's actually quite common from further research, that robins, if there are no eggs in the nest, they might accidentally go into somebody else's nest, just kind of chill out. So the blue eggs
blind and stupid.
They're like suicide cardinals, but not as cool.
So robins are stupid and blind. All right, that's why they have blue eggs.
So, once you complete this project, where does that leave you academic-wise? And what are your goals academic-wise once this project's over and you pursue other projects?
So I'm currently a junior in high school and a sophomore in college because I'm doing early college. So by the time I finish this project, I should already graduate college with my bachelor's, and I will have already graduated high school with my associates. So that'll lead me out where I'm academically. I'm probably going to go into the medical field for obvious reasons. And then project-wise, I guess I want to further pursue selective breeding of the birds to make them even more, and possibly even try to branch out into other non-avian theropod lineages that look similar. So like the Velociraptor fibula section that I brought up earlier. Do you know velociraptors were only about the size of a small turkey?
Yes, yes, I
They're
did.
tiny.
And the Velociraptor in Jurassic Park
actually
was
Deinonychus.
actually, yeah, that it's not actually a Velociraptor.
Yeah.
I like destroying people's days with that little tidbit.
And then so moving out to smaller theropod species like that, and then going for scientific accuracy. And so I guess one of the biggest questions that we've gotten on the Discord is how do you make something that's scientifically accurate if it's not around today? And then so one of the things you have to go off on are fossils. And then you say, well, how do you know what an animal looked like if you just have its bones? So fossils are really good at preserving feathers, especially in mudscape. So do you remember earlier when I said the Lemosaurus name literally translates to the mud-covered lizard?
Mud lizard. Yeah.
Yeah. And then so since they had feathers and they were preserved in mud, their feathers are extremely well preserved. So we can get exactly what colors the feathers were, everything of the sorts. And then we can even pick up possible feather patterns, which is how they got their fawn-like feather color that's now widely accepted for what they look like. And then so we can selectively breed for that. Also fossilized eggshells. Eggshells preserve extremely well, especially with whatever color the eggshells are. So if you look at, say, a T-Rex egg and an Oviraptor egg, they're blue. But if you look at a Ceratopsian eggshell, and a Ceratopsian eggshell is going to be whatever color the ground was because they're white. Whereas a T-Rex eggshell, some Tyrannosaurids had blue eggshells, and Oviraptor only had blue eggshells. So depending on the species, you can get different colored eggshells, even fossilized, depending on what colored eggshells their eggs were colored in life. And just so you know, it's not cross-contamination. Bilaverin, as far as I'm aware, can only be produced biologically. So it's not cross-contamination. It's legitimately what color the eggs were.
Wow, that's really cool.
Yeah.
So I guess making more accurate non-avian theropod models, and a couple of people inside of the Discord are like, when do we get a Triceratops? And so honestly, I don't know how to progress for something that walks on two legs into making something that looks like two legs to something that walks on four legs and has three horns on its head. The Trino, you know.
Start messing with a Jackson's chameleon.
Well, no, because no idea.
It takes time for science to advance to figure out how to do everything that we want to do. So, and as long as you're realistic with your expectations and like where science is currently at, I think you'll be good.
Yeah.
Brain farting. Never mind. I'll figure it out. If I don't remember it, I'll ask it in the Discord later. It was something about the Discord. No, I lost it. Where can everybody find your project if they want to follow along?
So I guess the best place to look is our Discord. And then so we post all of our content on our Discord along with, we also have a tab called Updates. Inside of Updates is a complete timeline from the start of the project for when we first started doing injections up until 3 days ago. No, wait, today is Sunday, so that would have been 5 days ago when we did injections for GFP control. And then those birds will get, they'll get frozen, then will get frozen, and then they'll be dissected. And then so the reason we're going for a week is, as earlier, just so ambient expression is no longer a thing, so we can gauge integration rates.
I just pulled up the Discord page. Are you guys seeing this?
Yeah.
Yes.
Yeah. Okay.
So then we have our first
here's all the
knockouts.
updates.
Yeah. At the top, those eggs incubating our first myostat knockout quail, underneath with them hatching. And then we have chimeric pigeon embryos that were produced using one of the methods by our friend Tom. That's with GFP right there. That's the glowing fluorescence. Do you see how part of the embryo doesn't glow and other parts do glow?
Yeah.
That's what chimeric is you're gonna have some of the cells that are modified, others that aren't.
Okay. What kind of incubators do you guys use?
So we have two Arcom King Suro 20s.
So big, fancy, expensive incubators.
It's entirely automatic. Besides, I have to switch it over for the last 2 days to increase humidity.
And this is the GFP quail?
Yeah, so this is one of our GFP chicks. Sadly, this one died of exhaustion shortly after hatching, and I didn't want to risk any of our other birds taking them out of the incubator because it hadn't even been 24 hours yet. So I removed the dead one from the incubator and I put it under a black light just so I could take pictures without harming any of the other quail, because as I stated earlier, our birds are our #1 priority.
Yeah, and the reason that you can't take it out of the incubator before the rest of them are hatched is because it can cause a humidity drop in the incubator and causing the internal membrane of the egg to shrink wrap to the quail, trapping it inside and therefore killing it inside the egg.
Mm-hmm.
And we want
that was
all
the
our
first one to
babies
hatch too. So
it's always the first one that's like hit or miss, whether it's going to make it or not, depending on how early it hatched out. So it's that's just kind of a thing with quail. So they don't go on your time.
I was kind of bummed. But that's actually the one where we collected the leg from for skin biopsy samples for PCR confirmation.
Mm-hmm. Tiny little thing.
Yeah.
So either way, like, yes, it's unfortunate that the bird passed and didn't make it, but obviously it's continuing to be used for
the project,
its purpose. Yeah, exactly. So, its life still had value.
Well, with any project, really doesn't matter if you're genetically modifying things or just breeding like we do. You are going to have some loss. It's just a matter of the fact.
You can't create life without creating death, unfortunately.
Yeah. And I mean, even if the eggs weren't modified, we were still going to have dead ones too. I mean, that's just quail in general.
Yeah, exactly. Like, and I'm jealous of your 80% hatch rate. So, like, that's a fantastic hatch rate for, anything, and you're modifying it as well. So the fact that you're modifying it and you still have a fantastic hatch rate, you're not modifying anything that has to do with the hatch rate. So obviously what you're modifying isn't negatively affecting the animal. So
I mean, you were saying? Sorry, I cut you off.
Nope, you're good.
Did Brandon say something?
No.
Okay. I guess it was reverb. I'm losing it.
It's the AirPods.
For the tail injections, being we're using a virus and everything, we're going to have a lower hatch rate, and that's simply because it's not to do with the construct itself. It has to do with the actual injection process. When you inject the eggs using a virus, you have to essentially destroy the membrane inside of it, the air sac, to get to the actual embryo or blastoderm to inject it, and then you create a surrogate air sac after, which actually, if you go back to the Discord and you look at the pictures for the injected eggs, all the ones that are on the rack together instead of the many eggs together, where it looks like they have holes on top and they're sealed with paraffin.
Come on, open up. That's these ones right here.
It's loading. Yeah, those ones. So if you zoom in, you can see on the top of each egg right there, that's where the air sac is, or off to the side of the air sac. And essentially, you go in through the air sacs. You're not disturbing any of the internal membranes of the yolk and essentially bursting the yolk and destroying everything. You go in through the air sac, and you can directly penetrate either the blastoderm or the embryo itself. The blastoderm itself is entirely encased in a membrane bubble that itself is separate from the yolk. And then it has enough nutrients inside of the blastoderm to get it to the point where it can branch out from the blastoderm and start feeding off of the yolk.
Science. You okay, Brandon? You okay?
Yeah.
So with you having to break through that membrane, is there an increased risk of bacterial contamination, even though you're still going over it with paraffin and sealing it back off?
So we do, first of all, we're doing this all inside of it's under what's called a fume hood, a laminar flow hood. Sorry, laminar flow, my bad. BSL2 grade. And then so essentially all the air pushes it away. And also it has UV sterilization. Obviously the UV sterilization isn't on. We don't want to affect the quail embryos, but essentially it pushes all the bacteria away from the egg. And then even then, because, really sketched out, we're using antibiotics and antimycotics. We're using penicillin streptomycin mix. And then I can't remember the name of the antimycotic we're using. So essentially that stops bacterial growth and fungal growth. So it gives the birds the best chance. The only thing that's the issue that causes a lower hatch rate is essentially humidity getting in through the surrogate air sac. That's it.
That's fantastic. Like it really seems like you're trying to cover all boards with this. And like you're really thinking of everything.
Yeah, I mean, I just want to make sure that that everything we do has the best chance, not only for the project to succeed, but I keep talking about it. I love these little guys, I really do. And I just want to give them the best chance that they can at a life. And I know I'm modifying them. I don't consent that I consent to that. But, they also don't consent when I steal their children and, eat them for breakfast, or when Darlene in the Facebook group cuts them open and then eats their organs because it's an old German recipe. So I don't have an ethical issue with modifying them. I just know that since I'm modifying them, I feel even more responsible for them than I do before if I just have them as pets. So I want to make sure everything not only has the best chance at life, but I want to make sure that everything's going to live happy, healthy, and reproduce fine.
So after your project is complete, what happens with the quail? Like, are you going to start your own like GFP quail funny farm?
So, I mean,
that's what I would do.
The GFP quail, they're already going to be well established, hopefully, in the quail community by this point. But as for the dinosaurian quail, the lemursaur quail, I can't call them a dinosaur. Well, all birds are dinosaurs, so that's not true. I can't call them a lemursaur specifically, and I'm okay with that. I've accepted that. The reason why I can't call them, even if they're indistinguishable, say I make them perfectly indistinguishable. So you had a time machine, you go back in time. This is not going to happen. But say you had a time machine, you go back in time, you go to the coast of China, you pick up Lemursaurus, you take one of my birds, you put them together, they're entirely indistinguishable. They even try to mate, but that's kind of frisky. Even then, genetically, it's not a Lemursaurus. Even if by the modifications we're doing, make it genetically closer to a Lemursaurus, it's still not a Lemursaurus. And then so that's really where part of the second phase of this project comes about. And then so the second phase of this project, it's really in its early developmental stages because we're not even done with the first phase. And then so fossils like these, as proved by Mary Schweitzer in earlier papers, or at least partially proved, you can get somewhat preserved soft tissue out of them. Getting back to the Velociraptor fossil. So she was able to get preserved blood vessels and osteocytes, which are bone-forming cells, out of a T-Rex femur.
Wow.
That's cool.
A decade and a half ago. And then, so obviously DNA degrades too down. So these were just impressions of old cells, but that soft tissue is composed by peptides and proteins. But the issue with that is you don't know what's cross-linked and what can be read and what can't be read. They use mass spectrometry. And so this is still being scrutinized, being whether these are actual proteins or cross-contamination. But if they do turn out to be actual proteins, in theory, we could buy fossils. I'm just going to use this for reference. This isn't Lemursaurus, but we could buy fossils like these, take the peptides out, first transcribe them into protein, and then hopefully have something that's even more genetically close to a Lemursaurus. But I guess that's for the second phase of the project for this, to get something that's closer. And if I can get something that's at least 10% for sure Lemursaurus, I'd be happy with the project. I'd call that a success. Not only would it phenotypically look like it, skeletal morphology would line up, but if it was even genetically more closely related than the starting base, that's a success in my mind.
Yeah, absolutely. Like, I think there's almost not necessarily limitless options to do for further research with this project, but like you could definitely carry this on for quite a while.
Absolutely.
And just check out what the next application is.
Mm-hmm.
How many people are involved in this project?
So if I want to go over members real quick, we have myself personally, we have Andreas Stumer, we have Raul Palmares, and then we have David Costello as a bioethicist advisor, and then we have Tom Bishop, who works with UC Davis as not only an advisor but also lab technician.
Okay, so about 5 people.
And then everybody in the Discord gives us our support.
Is everybody, there at the lab working together, or are people all over the world working on this project?
People are all over the world working on this project. Sometimes we'll meet up in person, certain groups of us, but as for Andreas, he lives in Europe. And so other people like me and Tom, we're planning on doing lab work together in the next oncoming months. Raul is studying up in Chicago right now for law. And then I do lab work, Tom does lab work, Raul does legal work. David, I've never actually met David personally in person, funny enough, but he does advise us on a lot of the ethics for the project. So certain parts of the project that we wanted to do, we didn't do specifically because we thought it wouldn't be ethical. And I feel like some of the reason why I'm so happy with this project, because everything we do is ethical, at least scientifically speaking. None of these things are going to affect the bird. None of these things are behavioral modifications. None of these things will affect how the birds reproduce or exit the egg. So that was actually a question of Discord, like the other day. Are they still going to have the egg tooth?
That's a good question.
And then so
Yeah,
it is a good question. So even
well, a while back, I was wondering if in like mainly the ball python hobby, most breeders cut their eggs. So I was wondering if long term it would affect them on an evolutionary level, and they would start to evolve to not have the egg tooth and just become reliant on us, essentially. Yeah.
So okay, so here's the thing. Their egg tooth, as of right now, is dominant. It's been dominant because it's been selected for millennia. And then so if there was a group of breeders and one of their snakes, say Brandon up here, say he got into ball pythons for some reason, he accidentally bred a brother to a sister, both of them were heterozygous for the egg tooth mutation, and now the baby doesn't have an egg tooth. If he cuts his eggs, that snake, even though it would die in the wild, could persist. And then if he keeps line breeding his lineages, that egg toothless snake could persist. And if he starts sending his snakes out, sure, I mean, you get small groups of them, or even say one morph. If a morph was descended from these egg toothless snakes, all of these snakes could not have an egg tooth. But I can't see in the foreseeable near future that all ball pythons will lose an egg tooth simply because we're cutting the eggshells. You might have individual cases or even small subsets in a region, but it wouldn't make sense if there's no reason for them to lose it as a whole.
Thank you. That's all I've ever wanted. Like, I think that explains it pretty well and makes sense.
Oh, absolutely.
Thanks for answering my question.
But to answer the Discord question about egg tooths, alligators have egg tooths, snakes have egg tooths, lizards have egg tooths. Most of those things I just named have snouts. Turtles have egg tooths. Birds have egg tooths. T-Rex had an egg tooth. Even if we give it a snout, which is an ancestral snout, let me make that clear. It's not like adding in another gene. It'll still produce the egg tooth, still be able to pip, everything will be okay.
Cool. I saw one of the other issues someone asked was, what happens if they escape? Which I know the answer to this, but
It's going to be Jurassic Park all over again.
but it wouldn't.
Actually, so two things. First thing is, this is why we can't get out adult birds. because the FDA and USDA, and I feel like they're a little bit too strict, but can't talk shit or else I'm gonna go to prison. So the reason why they're strict on the policy isn't only because people are scared of transgenetics, even though that's a large part of the policy. It's because if these animals get out, especially with crypnorids, so like the glowfish species, which are actually having an issue with this in Florida. In Florida, I'm going to touch on this and then get back to the main topic. Do any of you breed fish?
Nope.
I do not.
Okay. So in some fish species, especially in South American fish species, a fish that is more red is desirable to females because the more red he is, the more carotenoids he has in his diet, so the more territory he controls. So there's sexual selection for males that have more red on their body. And then so there was already a feral population of these tetras inside of Florida and from Miami, from people that didn't go from the pet trade. And then so when Tetra, which is a company, also the name of the fish, produced a glowing variant of these fish that was red, and people let them go into the feral population in Florida, females saw a fish that was entirely red. In every brain cell in their body that wanted to do sex was like, hey, this guy's got like a little red giblet on his fin. This fucker is entirely red. And then so now the whole population has shifted, at least in a large group of areas, from wild type fish to all of them being selected for GFP. Well, it's actually RFP inside. So then these feral populations, they had other varieties that were mixed in. So like long fins or other things or skirts. And then so when those wild populations saw the RFP variant, which is for red fluorescent protein, just like GFP stands for green fluorescent protein, when the wild population saw the red fish, they started intermingling. Now the vast majority of this feral population is all red. They're all invasive, so it's all technically a felony, but the FDA and USDA are worried about it.
Okay, so they're worried about your quail that only glow under black lights becoming more sexually attractive to the wild quail that
don't live on
the
this
wild
continent. That's what.
Okay. Stupid people making stupid laws.
So,
if my quail escaped, they would die.
Yes.
They would be dead. They wouldn't live. I have one California quail, and even if he escaped where I live, he wouldn't be able to survive. Now, they are invasive in British Columbia, which is to the west of us and just above California. So, like, I at least understand why they're invasive there. But they have a totally different climate than we do.
I don't think it's necessarily just the escape factor. It's what if somebody breeds them to their regular quail, and then all of a sudden, a couple generations later, nobody is known about the genetic modifications, and then it just becomes rampant and takes over.
But the only genetic modification is the GFP.
Yeah.
The only way you're going to even notice it is if you put a black light on it. It's not going to affect the animal in any other way than glow if you put a black light on it. So what's wrong with his quail breeding with my quail? What's the issue?
I think it's just the fact that, I think it's just the fact that people won't know about it.
Yeah. As for the dinosaur quail, I mean, it's the same thing that's going to happen as if an oil spill happened. I know you're like, how does an oil spill pertain to transgenic quail? Or these aren't even transgenic, they're modified. How do those two things pertain to each other? Well, if an oil tanker sinks in the middle of the ocean, we have an oil spill, the company has to pay reparations to all the surrounding countries that are affected by the oil spill, and then pay a fine to the United Nations for the oil spill, simply to help with cleanup efforts and everything. And then they have to clean everything up and hire everything to fix that. If one of our birds escape and we can't find it, it's most likely going to die. Especially where I live, there are no feral populations of Turnix japonica or anything in the Turnix genus. nor is there anything in the family that my quail are in that can hybridize besides turkeys. And turkeys are known to kill domestic quail populations for territory. So if one bird gets out, that's kind of reckless on me, but nothing would happen to the population in the wild. There's no feral population here. If, say, a dozen got out, or 100 or 1,000 or whatever, then we would fall into the same jurisdiction that an oil spill falls under for biological cleanup, containment, and everything like that. And also have to pay a large sum of money as a fine, and quite possibly could face prison time for recklessness and biological endangerment. That's it. It's no different. Yeah.
Even though it wouldn't cause that kind of damage.
Yeah. I mean, it's the same thing if Brandon went down to Everglades and brought a shit ton of albino Burmese pythons and let them go. He would get the exact same charge.
Oh, okay. That makes sense then.
Yeah.
Okay. Never mind.
Technically a biohazard.
Yeah.
Well, it's like anyone releasing captive animals into the wild. I think it's basically illegal everywhere.
Yeah.
Regardless
If it's on purpose or on accident, you're still responsible for it.
Yeah. I've had one quail escape and then my dog caught it in his mouth. And it lived. Something went into Pibble's mouth and lived, Brandon.
It's amazing.
It's beautiful.
Well, you guys want to wrap up?
Yeah, this was a lot of information and a bunch of stuff that I learned today, so that's great.
I'll teach you how to set up Discord, Brandon.
I probably won't use it.
I'll send you.
You can still join to
update.
boost my numbers.
I think we could do that.
Check out the Patreon in the description. Check out the Discord in the description. Check out our Patreon and like us and all that other shit.
We don't really do a whole lot on our page right now because we have like a whole 3 subscribers.
He is too.
I have 3. Thank you guys for having me. Everybody that's watching, specifically because I was on, make sure to like and subscribe. You know, the whole shebang. If you guys are interested in their podcast, make sure to tune in for more episodes. And make sure to support them on Patreon. They do stuff like this every week, or at least try to.
2 weeks.
Every 2 weeks right now. So.
Yep.
2 weeks.
Man, I need to get you on the show again.
Yeah, like we're trying to bring the academia side of the hobby to the hobby.
The public, you know?
Yeah.
Because there's a lot of things that the regular Joe Blow who has 2 ball pythons and a crested gecko doesn't hear about, and stuff that we don't hear about, and this gives us the chance to bring that information to the masses.
We just want to share what we're learning with everybody.
Yeah, exactly.
Yeah.
Awesome.
Educate the populace.
Yeah, exactly. It just puts information out there, you know, and like when two things I love kind of cross over, even if it's a small crossover, I definitely want to mash them together and do a podcast about them.
Well, thank you guys. You guys have a great rest of your day.
You too.